Okay. Reshare my screen. I think last time I actually forgot to record, but hopefully we get better and better. Okay, so first I wanted to go through just a couple of quick ground rules. That our primary goal for this journal club is really to build the longevity community and learn together while discussing cool science. So hopefully we've all um, at least tried to read the papers before coming so that we can have a really nice discussion. So if you have a comment or want to ask a question, you feel free to use the raise hand emoticon. And then when you're speaking, please introduce yourself because we do all want to kind of meet each other uh, and grow the community that way. And then if you're not speaking, try to avoid interrupting each other just to enable the best discussion. And then it's okay to disagree, but be kind. And then there are no real bad questions, but maybe try to not get tangled too much in the weeds. And again, this is like discussion based, so hopefully reading the paper and come ready to discuss. And then I'm also really excited to introduce you all to Dr. Amin Haghani, who's joined us, he is a senior computational scientist in the Horvath group at Altos Labs to help add to the conversation um, around this paper. And so we'll go ahead and jump right in. Um, so the paper that we chose for this first journal club this year is really about using serum or yes, so using plasma proteome in order to have biological clocks to think about biological age, which is um, an age which is a level of age-dependent biological changes like molecular and cellular that might be affected by molecular and cellular damage. But biological age is certainly somewhat different from chronological age, which would be just like, oh, you're 40 years old or whatever. But the age deviation is this difference between your biological age and your chronological age. And this has kind of gained a lot of attention as possibly being a biomarker for different age-related diseases or mortality risks. These aging clocks originally started by DNA methylation, but of course they've transformed and gotten better and better over the years, and also been applied to transcriptome as well as proteome. So I just want to see if anybody who's able to make it has worked specifically with any type of aging clock, whether that's DNA methylation or otherwise. Feel free to raise your hand. Would love to hear from um, anybody in the community who's worked with aging clocks in whatever context.
Oh, I have, and I think also maybe some of the authors of this paper are on the call,
Awesome.
actually. So,
Yeah, hi.
Yeah, Ludger, you're the first author, right? How are you doing? Good to see
So
you.
I'm good. Yeah, so it was being discussed. So I thought I'll just listen in and see what people think of it.
Okay, well, you have to at least admit you've worked on aging clocks.
Yeah.
Yes, yeah, I did, yeah.
Awesome. Thank you so much for joining.
Yeah, I wasn't sure if I was allowed to join or not, if it was appropriate or not.
Oh my gosh, of course.
Okay.
and I think
Thanks.
it's fantastic to have you here.
Okay, thanks.
All right. So moving forward, of course, aging clocks have lots of utility, whether that's in thinking about disease prediction and management or intervention and thinking about personalized medicine. Would love to hear thoughts, either really from anybody, on whether they think that aging clocks have more potential in one space versus others, or maybe you think that maybe they're not useful in different types. So I don't know if anybody has some opinions of where aging clocks probably have the most potential.
But I can comment on
Yeah, I
this.
mean,
So anything chronic diseases that age is a risk factor, like it's just about because the idea is to regressing the biology on age, right? So it's like so we figure out which part of the biology relate to age and which part of the health. So if age is a risk factor, then the epigenetic clocks, or actually the biological clocks, supposedly should have a relationship. But of course, it depends on the uh, again, it's like depends on your training set. How you train it. It's like you can decide what it wants to relate to, really. It's like the person who trains this. So it's like which aspects of health or which aspects of biology does it wants to capture. So it's not like all of them are the same, they are different.
mhm, mhm. Definitely being mindful of which clock is used in which setting seems important.
Yes, exactly. Exactly.
I have thoughts on this too. So there's a lot of debate on the, and of course I'm biased because I've been studying aging clocks for my whole career. But um you know, there's a lot of talk on the internet of aging clocks. They disagree with each other, and you know, how can you, and you know, I sent my samples off to Clock Foundation, and I got all of the clocks. So there's 6 or 7 clocks in Clock Foundation, and I was 22 to 52 or something, depending on which clock. Um, you know, but that actually I think is fine because they're built on different things, right? So Grim Age is built to predict how long you live, you know, age is built to predict, you know, your disease risk of various diseases. And so I don't think people can expect them to give you the same answer when they're defining aging in different ways. So I think part of it is that there needs to be better explanation of you know, how people are defining aging and what the clocks are useful for. So I think that's kind of gotten lost in the messaging because people are expecting that there's one biological age, but there isn't one biological age, which of course this paper is telling you that there's different ages for different organs, which is definitely true. And then I would say there's even different ways to measure the different ages and different organs. So, you know, I think there's a lot of nuance that's getting lost in this, and people are throwing the baby out with the bathwater instead of interpreting these in the correct way. My
By
two cents.
people, do you mean the general public or scientists? What are we talking about?
Influence science influencers on the internet that
Mm.
are I have big opinions on this. I
Mm.
mean,
Yeah, they
in
are
some
super skeptical, which completely makes sense because there is disagreement between scientists. So how do you expect like the general consumers understand what's going on?
Yeah, also, you know, I get it. You know, if Halle Berry goes and gets, you know, glycan age and gets a younger age and then goes and takes a true age and gets an older age, she's gonna be annoyed because which is the real one,
Yes.
you know. So I think there needs to be some way of just explaining this all better.
Mhm.
Um,
Where do you think that onus
Yeah.
lies? Is it more on the physician who to like make sure, or like say the family practitioner to make sure that that particular patient is interpreting that data in the best way? Like I think about just like genomics and where that came from, and encouraging people to not just like totally trust their 23andMe or whatnot, but to like really rely on their own physician to interpret that? Or is the onus on the companies to make it super clear where the limitations are for their particular model, or really everyone?
Yeah, I'm not really sure how to fix the problem.
Mhm.
I mean, I can tell you for me, what I do is I'm like, okay, my average biological age across all of these is younger, so I feel good about it. I'm just taking like a mean, but I don't know if that is realistic for people to do. Or you pick one and you just follow that one and you know what it's measuring and
Mhm.
you know, I think ideally maybe we just have a bunch of different clocks and we know what each of them does and we
Mhm.
follow them and there's a in-depth explanation, but they're kind of too expensive to do a bunch of them at once usually.
Yeah.
So I don't really know what they is, but it's worth trying to figure it out how to fix
Mhm.
So actually, the truth is, whenever the field is a chaos, and that's will cause all the confusion, and that's why actually the biomarker of aging consortium formed, so to come up with a standardized way to benchmark things, and that completely makes sense. In any field of science, when there is chaos, the kind of consortium needs and actually the consortium needs to be formed to come up with the standardization and kind of benchmarking thing. So otherwise, it's like I don't know if you've seen the recent podcast about the person. It's like one of the Washington University PI. He questioned all the clocks. Actually, the public is so skeptical now about even the applicability of any of these because the guy sent his sample to 8 different companies and received and to some of them multiple times, and each time the age was different. And like then they said like these are meaningless and the scientists are supporting this. Like why are they putting their name on these
it.
companies without knowing this? So
I think that was Matt Cableines, wasn't it?
yes, yes.
Yeah, which was I thought quite good. I personally I've given up on the methylation clocks as they don't provide useful information. I think in the end you're looking at functional tests. And one of the difficulties is that a lot of the methylation clocks are based on white blood cells. Now, does I have my own theory as to what aging actually is, which is based around the efficiency of the mitochondria and senescent cells. But if each cell has a different age, potentially, um, what are you going to do? You can measure what Matrix Bio do is they measure mitochondrial DNA damage in kidney cells that come in the urine. Most of the clocks, methylation clocks, measure the white blood cells. Um, Now, are the white blood cells are important because they spread mitochondria around the body, but are they an indicator of any other sort of cell? If you take buccal cells, it's all sorts of things. I've given up with the methylation ones, though. They don't provide actionable information.
Actually, I will disagree with that, but they have a lot of information. Of course, I don't think my it's like
Actionable information.
actionable is not the problem of the clocks. The problem actually is the intervention field. So it's like that's a big issue. So it's like still we don't know what is rejuvenation to come up with a actionable
Well,
thing.
I have
Yeah.
my
So
own views as to what rejuvenation is, but the fact is, in the end, if you have a measurement, you need to have some idea of what change of intervention you're going to make from that measurement. And a measurement that just says, oh, I'm actually 64. My true diagnostic figure came out at 37 at one stage. That's very nice. But what does it tell me about the interventions I use? It doesn't tell me anything at all because these things
no,
go up and down.
Well,
no,
so
there are many of these clocks are reversible. So I
yep.
mean, it's like there are interventions that show this. So it's like that's where the action comes in. So it's like if you
Yeah.
understand the biomarker and you know it's reversible by through an intervention, then you can try it out. Like for example, exercise or like even some of them
Yeah.
are sleep routines. Like simple things can reverse the clocks back.
Yeah.
Mm,
Well, we disagree, but we don't have to agree.
Yes.
Awesome. Well, let's go ahead and dive right into the paper. I just wanted to quickly introduce also the UK Biobank data set used in this paper, which is an amazing data set that's huge that includes around 500,000 UK participants. That also includes de-identified information about genetic and lifestyle and health information. And so it's one of the most comprehensive and widely used data sets because of how expansive it is. And in 2023, Amgen was able to do plasma proteomics for this database. And so that is the data set that was used for this paper to dig into plasma proteome clocks. I don't know if Luke, you want to add anything to that and like why UK Biobank data? I know it was kind of like you know, one of its kind, and also there were additional data sets used in the paper as well that I think was a really good call. But if you want to add anything about like why this data set.
No, I think you covered it quite well. I mean, it's such a large data set. Data was available, so it was like, a logical next step to try to use UK Biobank. And I think the second thing that's new in the paper is also the mortality-based clocks, but maybe you're gonna touch upon that. And so
Totally. Awesome. Well, let's get right into it. Of course, thank you so much for joining. And then
thank you for discussing it. It's really I feel honored.
Yeah, it's a great paper. And I just wanted to point out that a patent application was filed from the paper, and then, of course, the affiliations of the authors. So one of the hypotheses that was posed within the paper was, are chronic diseases manifestations of aging? So kind of keeping this question in mind as we go through the paper, and we'll kind of come back to this as well. But first, this paper using the proteomics data set, we're going to go through a couple different models that they used, and then would love to get thoughts from people who I'm certainly not an expert in this field, so if I get something wrong, just tell me. But first, they use a first generation chronological aging model where the UK Biobank data sets were split into about 5 equal size, non-overlapping test sets. And then after training, they found that this worked pretty well. This worked very well on the test data sets where they saw a nice correlation between the predicted age and the chronological age. And you can appreciate here how even for male and female patients, this worked very well. I did want to point out that you might see patients like this that have a larger age deviation where the predicted age is much higher than their actual chronological age. And you can see you have multiple patients of that type even in the training data set. And here they use this elastic net model, which is really useful for these large data sets. So from this first model, they got good performance indicators. There were no sex differences in the age deviation, but then this also was able to show that the predicted age correlates negatively with grip strength, which is a appreciated, which is a well-known thing. And so then from here, they moved on to think about this mortality-based model, where the chronological age was not used at all for the training of the model. And yes, I mean,
the question from by the way, how do I pronounce your Luger?
Yeah, that's fine. Yeah, I say
yeah. Yes. Okay, so Luger. So did you adjust for cigarette pack years, or like how did you deal with this?
No, this was purely trained towards the outcome. So it's either towards chronological age or towards mortality with a Cox proportional hazard elastic net model. So no adjustments for anything, not even sex, just trying to predict those things based on the proteomics. That was how the models were trained.
Okay, good. Interesting. So but the pack-care data is available in UK
Yeah,
Biobank,
yeah,
right?
that's available. Yeah, at least for I mean, for people who used to smoke, at least for a subset of them. It's
Okay, cool. Because I think like maybe male and female, if you adjust for pack years, there might be differences because they respond differently to cigarette smoke really in environment.
Yeah, I mean, there's a lot of exposures which are available in UK Biobank, but the aim here was really to kind of train a clock that could either predict chronological age or mortality. So I didn't want to make it, I mean, there are millions of confounders that can affect biological age. Many of them may not even be measured. So the idea is not to train a smoking clock; it's like an aging clock, and then afterwards you can see does pack years of smoking affect this predicted age? That was kind of the idea, or in different organs and also.
Yeah, makes sense. Thank you.
Yeah, I think it's great. And even here you can start to appreciate the sex differences with male and female patients. And that's shown even clearer here. Where yeah, of course, where for the mortality-based model, chronological age wasn't included yet, there was a strong correlation between the predicted and the chronological age. And then they also they also saw sex differences with the mortality-based model. And then one thing that I thought was really nice is that they also used additional validation data sets, such as the multi-ethnic study of atherosclerosis on this model, and found that it generalized to that model as well, making it even stronger. Um, so, and then, so then the paper digs into kind of what proteins were drivers of this, and then they found that from about 3,000 proteins that were measured, 1,664 proteins were significantly positively associated with and 665 were negatively associated. And here it was interesting that different proteins influenced either the first generation chronological model, and then really a different set of proteins, or very different looking set of proteins, impacted the mortality based model. So you would have different biological markers influencing aging versus mortality.
Something about this one also. So this analysis is actually more like the statistics. So it doesn't have to do much with the models. It's just like each individual protein, what is the association with chronological age or the association with mortality. So they're not necessarily drivers of the models, even though, of course, proteins with big effects will be important in the model. And also maybe some people might wonder why you see so many significant proteins. That's because UK Biobank is very big. So even very small effects are gonna pop up as significant because you have so many participants.
Mm-hmm. Is there a reason why there's more proteins positively associated versus negatively associated?
I don't know. That I don't know.
Okay.
Yeah.
Um,
It might be inflammation markers. That would be my guess. That related to inflammation. That's what we see in the brain, at least. Yeah.
Yeah, that definitely seemed to be true with the mortality based model, which maybe makes sense more than the chronological model, where it seemed like there were more adhesion and secreted proteins that correlated with the chronological age model. But certainly a lot of immune cells, like unsurprisingly, are immune proteins or inflammation proteins. Yeah, and
they're not extremely significant as pathways, but those are the top ones at least. So it gives some indication. Yes.
Yeah. And then I guess like pointing, I think, Kristen, to that point as well, ultimately only 3,000 proteins were measured. It wouldn't be surprising if, like, I don't know, if maybe you could, Ligur, you could add to this, but I wouldn't be surprised if those 3,000 proteins were maybe even enriched in immune proteins, given that it's
I think so. The Olink has an immune panel, and that panel is also part of those 3,000 proteins. So yeah.
I like to comment, get everyone's opinion. So some people are skeptical in general about the Olink technology. Like for the actual protein measurements, they think it's like some of this might be noise. But of course, when it comes to biomarker, it doesn't matter as long as it's a consistent platform. That should be fine. So anyone has opinion about Olinks? Or I'm not experiencing Olinks. I just like this is something I heard. So eager to hear people's opinion about the platform.
I know it's antibody based, so it's a bit similar to, with the Western blot, you're also not always sure if you're only measuring this protein and so on. But indeed, for building a clock, technically it doesn't matter as long as you use the same antibody, because it's just a feature that is part of the predictor. Yeah, and I think, there was kind of a paper which kind of compared, I think, Olink to SomaScan also, but I think in the end it turned out, because there was some correction, and afterwards it turned out they're quite similar, but it's antibody based, so you always have to be a bit more. Yeah, you have to trust the antibody, of course. That's what I think, but in general, it's a very nice technology because it can be very high throughput. So you, with mass spec is also, I have a myself, I have a mass spec background, and I really like mass spec, but I don't think currently it's possible this kind of high throughput, measuring so many proteins and so many samples. So that's the nice thing about these protein array technologies, I think.
Yeah, it would definitely be expensive. It would probably run you $1,000 a sample to do mass spec, right? It's a, I don't know, 10x difference in cost.
Right.
But this is only measuring 1/10 of the proteome, right? Something like that. But yeah. There's the other thing to do with it, the whole inflammatory pathways. So this is the other debate. If the clocks go up and down acutely, does that mean they're useless because inflammation increases or decreases? You get temporary increases in the clock or decreases in the clock. Glycan age definitely has that feature where, if you're unhealthy for a couple weeks because you're traveling, it'll spike your glycan age and then it'll come back down. But I honestly think that that's like a feature, not a bug. You see the same thing with glucose measurements. You'll have some temporary increase in glucose, and that doesn't mean that measuring, long-term risk factors for diabetes is useless just because it acutely goes up and down. There's like another.
Yeah, I think it's also a bit of kind of maybe an unavoidable consequence of, like, you use a lot of features and then you try to predict a single value. So any kind of intervention that was not in the training data set, your clock is going to say either, that's what I think, it's going to look more like an aged person or like a younger person, right? It's one dimensional. You read out of your clock. It's just the nature of the clock. So if you have any kind of intervention that increases inflammation, and of course inflammation goes up with age, then you expect that your clock will predict you to be older. And if there's anything that maybe looks like a younger person as a molecular signature, even though it's not, it's a different state, the clock will predict you as younger, because a clock can only say older or younger. It cannot say like, you're younger and you're immune, but you're older there, and So it's kind of, I think, the nature of a clock. If it gives only one value, it's either younger or older.
That's interesting. All right, so then just comparing these first two models, one being the first generation or more chronological age-based model, and then the other being the mortality-based model, where there's more pronounced sex differences. It's using mortality risk in its calculation, and it provides these maybe more specific age-related insights, or age-related disease insights. So then, kind of taking these models a step forward, next they wanted to use these models to specifically look at organ-specific clocks. So in order to do this, they applied these models on proteins that were where they defined these as organ-specific if, according to GTEx, they were 4 times higher in one organ compared to others. And hopefully, or Luca, if you want to add anything like specifically about kind of how the organ-specific model is trained.
It's actually kind of the nice summary about it. So we picked GTEC. I mean, we picked this approach actually because, I mean, this was what Tony Viscoray had also done in a previous paper about organ-specific aging. So I didn't actually go too much in depth, like when I did the analysis, like different methods of defining organ specificity, because it's a bit arbitrary, of course, you could say. But actually, the second author on my paper, Anastasia, she also worked a little bit on this, and she looked at many different ways of defining organ specificity, also based on proteomics. And actually, this surprisingly, this 4x enrichment worked quite well if you wanted to predict, organ-specific diseases. Actually, this approach was one of the best, or maybe even the best. So it was. And the idea initially that we did it was because there was kind of Vadim's idea also that, like, if a protein is, let's say, produced in the liver, but then it goes through the blood and it ends up in the brain, then it's still a liver-specific protein because it comes from the liver. So that's why we used the transcriptomics, and we said, okay, maybe this GTEx transcriptome enrichment makes sense for that approach.
Yeah, I like that. That makes a lot of sense. So then, kind of like thinking also back to the original hypothesis, or one of the hypotheses mentioned in the paper of are chronic diseases manifestations of aging. I think that this like really comes back to that, where you can see the performance of both the first generation model as well as the mortality-based model, in that the liver model trained on these liver-specific proteins, the top disease is liver cirrhosis and fibrosis, and for the long model it's COPD. I think it's like maybe also unsurprising that for the kidney model, liver cirrhosis is up there as well, along with kidney failure. And then this is, the training set, and then the test set, and then this out of fold set. I mean, yeah.
So I have a question. Did you select multiple clocks, for example, liver clocks, and selected the ones that has the highest association with liver cirrhosis, or no? This is like you just trained it and it has the best association.
So the cool thing is that it's trained towards either, so the first generation is trained towards chronological age, or second generation or mortality based is trained towards mortality. So it's purely trained towards that. And then the idea is, can it predict disease? So it's the same clocks, same models, but trained on a subset of proteins that is organ specific. That was kind of that was the idea.
Interesting. By the way, your liver clock relate to kidney better than the kidney clock. So
liver, kidney. Yeah, it's possible. But there's also of course the issue that you only have 3,000 proteins in total. So in general, overall, not always, surprisingly, but overall, the conventional model is the best for many diseases because it's more powerful, you use more proteins. And liver is also a way bigger model than kidney. So there are more proteins included. So of course, if you have more features, you can also generate a better model. So there is this kind of thing also. The number of features makes, because it's organ specific, you have to reduce the number of features a lot. You can only use the organ specific proteins to train the model. So you inherently lose a bit of power towards, compared to the full model, or towards, organs that have more proteins, they may be a bit better at predicting these things.
Does the liver model work better than the previous model for these diseases?
Than the full model, the full model. I should check. I'm not sure. I think the full model is best for most diseases. Still, but not for
Okay.
everything. So I also, like in one of the very last figures in supplementary material is like an external data set with, how was the disease called again? Where you get like sarcoidosis, that was the name, sarcoidosis. And there actually the conventional model doesn't pick up on sarcoidosis, but the lung model and especially the immune model do. So they're trained on a subset of features, either predicting mortality or chronological age. And the conventional model doesn't see a difference, but those organ-specific models, they do. And this disease, sarcoidosis, is like you get like this kind of inflammatory nodules throughout the body. So it's really an immune disease, and it's especially severe in the lungs, apparently. So it really that made a lot of sense also. So it's often the conventional one is better just because it's bigger, but it's not always the case.
Okay, great. Thank you.
Yeah, and then to point out some other things from the organ-specific models, of course, here you can still appreciate that the kidney model, is picking out diabetes and kidney disease. But one of the interesting things that was also pointed out is that while the brain model, you see dementia being at the top, dementia is actually corresponds with decelerated aging of other organs. So I'm kind of interested if Luca or anybody has some thoughts on why that might be.
It's not decelerated aging per se, because it's after correcting for the conventional model. So what it shows actually is there additional information. So that was actually relating to the previous question. Generally, the conventional model is better. But then what happens if you remove overall age? Can you still see some additional information in those models? And then you see a brain. So even if your conventional clock predicts you're going to get dementia, if you have that information already, and then you use the brain clock, the brain one is the only one of all the organs which is going to tell you something extra. It's gonna, you're going to be older, so you're going to be more likely to get dementia.
Very cool.
So, and then you see this nice cluster also of all those metabolic diseases. I think you have lung with COPD, which is also in there. So they kind of make sense in that respect also.
Yeah, very cool.
I mean, I think there's also this issue that to get dementia, you have to survive the other diseases because it happens later. So
that's another point that Vadim also mentioned, which is indeed the dementia is a bit of a strange disease sometimes in UK Biobank in terms of how it behaves. It's indeed very likely also to do with this. So you need to be good or healthy in all the other organs in order to survive to the old age, in order to, get dementia in the first place.
Yeah, that's so true. The survival bias is definitely there. And then wanting to point out, I guess this kind of maybe illustrates some of the points that you were saying, that the conventional model might be better in some cases, or really in most cases. And then some of the other things from the paper I wanted to point out is that, there were, I think we've probably heard often across longevity sciences that men age faster than women, but I really appreciated that this points out, specific organs or specific organ clocks that demonstrate this, whereas others don't, which I think could be really important when thinking about patients. And then that of course individuals with lower biological ages have a higher age acceleration, and of course a higher age acceleration accumulate people with the higher age acceleration accumulate more diseases, which I thought was interesting and worth pointing out. So then to kind of summarize, just putting together all of the things that we've discussed, I did want to take some time to kind of go through some of the limitations of the studies and see if people have thoughts on maybe additional limitations Where I know we've hit some of these, even like protein designation. Luca, you were mentioning that this is antibody based, and so even if it's wrong, it still is useful for the model, and there's still utility in that, and of course the measurement technique limitations, and then UK, it's still the UK Biobank. It's still like predominantly participants or patients in the UK. And so then how generalizable this might be in other data sets. And at least like this was certainly gotten at a little bit by incorporating additional data sets, but there's always the question of generalizability. So yeah, are there any thoughts on additional limitations beyond what's mentioned in the paper, or Luger, if you have any additional thoughts, or I mean.
I think they're nicely listed also. I think with respect to the last one, like other data types for organ specificity. I mean, I know Anastasia is I think her paper is already on bioRxiv, if I'm not mistaken. So she investigated also this, and she came up with like a panel of 21 proteins that are then more with the aim of predicting diseases. But like kind of a protein panel that predicts most age-related diseases, but as part of that study, she considered a lot of different ways of assessing organ specificity also.
Oh, great. Should throw the paper in the Cool Papers channel or the Journal Club channel as a
Sure, I can share it. Yeah, of
yeah,
course.
that would be great. Thank you.
Yeah, welcome.
A question, can you comment? I'm seeing a plot that you compared the mortality-based model with the Grim age, and like which is epigenetic, but somehow it's like it looks like the hazard ratios of these proteins is much higher than these epigenetic based plots.
Yeah, I think there was kind of a small mistake in that plot, if I'm not mistaken. Wait, let me see. Because
I will be
I think in the final version, for some reason, because in the text it's written correctly. Because I have in the text it says like it's, wait, let me see. So it should be, I mean, the mortality based are all better, so they should all be better. But chronological ages should be relatively similar to the other ones. But it's also a bit so it's an kind of an approximate way of comparing, because you can it's like you have different studies, right? So Grimage was used in a different study, so it's a different population. So you always have to be a bit careful if you use this kind of comparison where you use like hazard ratios, if there's like a different population. But in general, if you look at the text for 3L, I think it's like so the first generations are, compared to O et al., our brain, intestine, kidney, and lung are better, and the others are worse. So it's really like very similar in range. And the mortality, they are all better, which makes sense also because the other clocks were trained to predict the chronological age. Ours were trained to predict mortality, so it makes sense that they're better.
Yeah, because I will be surprised if
so it is better than Grim age. Yeah, the mortality based. Yeah. Well.
Yeah, so you tested, you exactly use the same paired data of the same
but it's not, yeah, and it's also that's the other thing. Of course, it's an approximate way of comparing it. So I try to standardize the hazard ratios, but of course it's different populations. So it's also it gives some kind of idea. It was like a suggestion from the biomarkers of aging paper to kind of standardize the hazard ratios by the standard deviation of the population. But of course, if your population is different, I mean, it gives some kind of idea. It's a way to compare, but it's not, I mean, the best comparison probably would be to use the same population to take, from the same people, a methylation array, calculate Grim age, take the proteomics, calculate our models, and then really to the hazard ratio calculations. So it gives kind of an idea, but you have to be mindful that that there's some different population characteristics also. Yeah.
Okay, cool. Thank you.
Yeah, you're welcome.
Yeah, great. Yeah, so then just kind of wanted to take some time to think through either critiques or things that are yet unknown. And I know Mary and Vishal and I kind of were thinking through a number of these. Like we really appreciated the addition of other data sets and then, we were curious, taking from this paper, thinking about in the future, how could you use organ-specific clocks as into phenotypes in clinical trials or personalized medicine? So would love to hear thoughts on like, how this paper might be impactful for the future and how we can think about organ-specific clocks for actual patients. Or do people have other critiques or other things that they're curious about from building
I
off?
think it's too early to call this for patients. I don't think it will get any FDA approval anytime soon. But I think there is new government initiation to develop a biomarker for health. So it's like I think FDA is moving, but yeah, it will take a while, and who knows when.
So we actually do have we are actually in 13 clinics now with our brain aging clocks. Not FDA approved because we're giving people an age, not a disease diagnosis. Although we are running a clinical study right now that's funded by ADDF and Gates Ventures to get FDA approval. So I think they can be used now. Ours is combined with functional measurements and also brain MRI, so it's a little bit more robust because there's a bunch of different ways to look at brain specific aging. But yeah, I think these are going to be definitely adopted in clinical trials and in the future. It's my opinion. We do need to figure out, which clock for what thing and which one's the best and how do you even define the best. And, I think there's a lot of work being done around that.
Yeah, also an idea that I had, I don't know how realistic it is, but maybe in the future, you could have because if you measure all those, if you just measure let's say thousands of proteins, no doctor will be able to say, okay, this protein is really out of range, there's something wrong there. So I think this kind of composite biomarkers could also help point to maybe something is wrong. You have an overall clock, which gives your overall age, and then you have a brain clock, a kidney clock, a liver clock. And then if the liver clock says, okay, yeah, your liver is all of a sudden way older, then the doctor might want to know why. And then maybe you can have some sub markers, maybe this in the liver, this in the liver, this in the liver, and then okay, it's something wrong with that, and then you can really delve into detail to find some kind of diagnosis as to why all of a sudden this organ or this system is all of a sudden affected like that. I don't know. Maybe that could be some clinical application.
Yeah, I'm curious. Do you view the organ-specific models as disease agnostic or not? So I know that liver cirrhosis is high for the liver model. So then is the liver model truly disease agnostic as a liver aging model, or is it just a predictor of cirrhosis? Or could you add a little bit there?
I think they tend to predict. I mean, that's also what Anastasia then looked on later on. So they tend to predict quite a bit of diseases simultaneously. Of course, from a maybe bioinformatic point of view, I would think it's also since they're trained towards mortality or towards age, especially if you train towards mortality, they will. I would expect. I didn't go into detail, but I would expect that they would pick up, or they would be way better at, or maybe biased, if you call it like that, towards diseases that are really dangerous and that will somehow shorten your lifespan, and maybe also partially towards diseases that are more common. I would think if there are many people in the UK which die of a certain specific type of liver disease, then I would expect the model to be better at picking it up because in the end it's predicting whether you're going to die.
Yeah, and certainly it knows what's in the training data. Yeah.
Yes. Yeah. but the training data is quite extensive. It's like 50,000 people for which there is proteomics. So that's, I mean, that's quite amazing. That's also why initially you see those very good results, I think, because with many models like epigenetic clocks, you have like maybe 100,000 of features, and typically the data sets are relatively small, so you have way more features than people. Here it's the other way around. You have only 3,000 proteins as features. You have 50,000 people, so it's really I'm quite confident that we don't have an overfitting issue, and you see that also like everywhere. So the data, so that's really nice. You have way more participants than features.
Yeah, awesome.
Also, I think what is considered normal aging is really a continuum with disease. So cirrhosis is just the most extreme of liver aging, but whatever's happening before that doesn't mean it's not decreasing function of the liver, or so having this arbitrary cutoff of what's a disease and not a disease is, I think, part of like a misunderstanding of how this is working. We see that in the brain. You see neuron loss during quote unquote normal aging. You lose 10% of your dopamine neurons per decade. It's not till you lose 70% that you get a Parkinson's diagnosis. But that doesn't mean losing those neurons before then is good or fine. Or, I mean, it still makes people slows people down, makes their movements worse. I mean, so we arbitrarily make these cutoffs of, I guess, usually they're when it affects quality of life to the point that you can't live independently, or it's, you can't do normal functions, then we call it a disease. But really, all of this is pathological.
Yeah, that's a great point.
Yeah, that's part of the thesis of the paper also. I think like diseases as manifestations of organ aging, like the worst kind of outcome of a very aged organ, you will designate it as being sick.
Yeah, absolutely.
But at the end, any decision is dichotomized, right? So it's like doesn't matter. Even if we use these aging biomarkers and we say accelerated aging is a disease for like it could be tissue specific or non-tissue. It has to be cut off at some point again. Like that's the thing. So it's like we cannot. So
I don't know.
I guess
Why isn't it cut off at peak function? So most, for most things, your peak function somewhere between 18 and 25, depending on the organ. Why shouldn't we be aiming for that instead of not, not having it mean that we can't live independently? Why is the cutoff so late?
Because clinical trials will be so hard to do when it comes to say. So it's like how do we want to do clinical trials on 20-year-old people and monitor them for until they are 70 or 80? Yes.
Well, that's why you need surrogate endpoints, right? So the biomarker.
Yeah, it's really amazing how the field is rethinking, what is disease and, how can we measure things to really, I think it's just going to be amazing how the field evolves to really improve patient health and be able to identify diseases and redefine where diseases start, or, be able to catch things earlier. And I think that's where the longevity field, I think, is really certainly making a huge impact. Then kind of with the last few minutes, just wanted to point out that there are many, many companies, I know that we've touched on some of them throughout the discussion, that are thinking about aging clocks and utilizing clocks in many different ways. And I'm certainly excited to see where this goes, and to see where aging clocks start to be super impactful. And then just wanted to take the last minute to make a plug for joining the Longevity Global Community, and this awesome community built by Kristen. And that if you have any papers that you'd really love us to have a discussion on, definitely put it in the cool papers or the journal club Slack, or also just feel free to Slack me if you have a paper that you feel really strongly about. And then just wanted to say that, there are lots of different papers that we think are awesome in this area, and we'll share this PowerPoint so that you can maybe read even further into awesome work being done in the aging clocks field. So with that, I think we're right at time. If there are any last minute comments or anything like that. And thank you so much, Amin, and thank you so much, Ludger and Kristen, and for the awesome discussion today. I really, really
Thank
appreciate
you for
coming.
Yeah, thank you so much, Rebecca, for organizing this. What a cool event, and I feel like it was very successful. I hope everyone got what they were looking for out of it, but very cool.
Thank you so much, Justin.
All right. Thank you. Thanks. It was great discussions.
Awesome.
yeah, thanks a lot.
Yeah, thanks a lot, everyone.
Bye.
Thank
Bye.
you. Bye.
Bye bye.